Hi again. We're going to cover joint variant calling now. Last time in our hands-on exercise, we covered using haplotype caller for individual samples, for looking at the calls, examining the BAM files, examining some of the internals to what haplotype caller was doing when it realigned some of the soft clipped reads, and basically seeing how we can get more confidence in calling at the single sample level. We barely touched on joint calling when we used genomics DB import last time, but I'm going to focus much more now on that process. So in the general best practices pipeline, we're now in the middle part there. Okay, we had our raw mapped reads, we've mapped them to reference, we produced our BAMs, we marked duplicates, we maybe did some recalibration, and now we can go in and produce some GVCFs and actually start calling together. So when we talk about joint calling, we're in the middle column. This is a slide you'll see a lot, by the way, while we're here. So as Eric mentioned before, the real thing is you want a joint call because it gives the variant calling tools much more information. Again, better contextual information means better confidence and better calls. And that's our goal ultimately. Not just to say, you know, I got a value there, but to have a value you're really confident in. And it really helps us know, and also helps us know, even if we make a particular call, whether or not that call is actually common or in context. Is that a rare call? Is it 1% of the population, 0.1%? Is it unique? Is it a de novo mutation in that one individual? You don't know that looking at it by itself, but you can know that by doing joint calling. And the general process that we see is that, you know, we produce GVCFs, we bring them together somehow, and then we can do our analysis. Now, the thing is, it's also possible in joint calling that we can actually make calls, even at the individual level, that we couldn't alone. Like, because you have evidence across multiple reads, like in sample one, okay, the reference A, you got 2 G's there. Was that an error? I mean, I don't know. All by itself, you might look at that and say, well, I don't know if that's strong enough. That doesn't seem enough to really call that as a variant. And maybe you got another sample and they've got a bunch of A's there. But you also have other samples which have a couple G's there. Well, if you got a couple of samples that seem to have weak reads on the G there, but otherwise the error is being sequence findable, then that actually gives you reasonably confidence that maybe those G's aren't just artifacts. If multiple people have weak reads on the G together, that tells you something about that G. You can call it. Maybe not with 100% confidence, but with enough confidence to say this is something we should not ignore. And also it's important to actually remove some various biases we have in there. So how much I'm going to go into the same bias? So sample one, right? You got a whole bunch of reads here at a given location. Let me use my second here, right? And again, you see some C's among the A's, and you don't 100% know. You don't really know what's going on. And you see the forward one here. You see in reverse. Okay, well, it looks now, you only see the C's on the reverse part here. You only see them there. Now, is that an issue here? Again, if you have a lot of samples together, you might be able to see, maybe I got a strand bias going on here. Maybe if it's only showing up in reverse, that shouldn't happen. In theory, you should see it going forward and reverse across there if it's real, right? But if we happen to see we have a strand bias at that location and we're only seeing an allele most of the time in one direction, that also gives us confidence that, you know, maybe that's just noise. Maybe that's just something going on at that location with the sequencer as opposed to something real. Like I said, more information lets us actually know that. And again, as we see here in one sample, we can say, all right, right here, this allele shows up. You have some C's showing up here and a bunch of A's, but less of them showing up on the other side. So if you're looking at one, do you really know? But again, if you look at them and over here, you can say, well, all right, in this case, I have the C's only showing up, like all of those C's are only showing up on back reads and the A's on the forward reads. You look at that and say, regardless of the number, I can see that now. I can say if they're only showing up one way, that's an artifact. That's an artifact of when we're just reading that direction. That's trend bias. But even if you have the same number of A's and C's over a large enough population, you can see that actually it's showing up. It's showing up forward, it's showing up backwards. Statistically, we're seeing it on both strands as we should. You can call it again. Might be hard on one, but when you're looking at a population, you can filter out certain issues you had with sequencing by saying, statistically, how often does it show up and how? And there are a couple of different annotations that actually help there. So, for instance, when you look at just one sample or a couple, that you can actually see that there's a distribution of, for instance, common variants and the rare ones. It peaks here, and that's actually okay for a good one. But when you have a lot more samples, something that looks like it's rare, it looks like it's peaking, those will start to actually even out some. Like you can actually have them start, if it looks like it's rare in one sample, looking at a large enough population, it might take on the distribution we'd expect to see. Like when you're looking at it, again, in Nomad, for instance, you can see that shows up, but it shows up looking like much more of a common one. When you have enough people in there, you can start to actually get better distributions and say, well, okay, that actually makes sense. Over here, for the ones that appear a little further from zero, they're likely to be junk. And when we look at the algorithm that help us filter good variants from bad, if it's actually good, if it's actually tight, if it's actually near zero, we have a lot more confidence in it. Well, again, looking over populations, we can say, here's what the variant actually looks like. And okay, that was not junk. Even if in an individual sample or just a few, it looks like it is. So the other important thing is that when we measure our ability to, we measure how good we're doing in joint calling, there are two things we look at. One is sensitivity and one is actual precision. Sensitivity means let's look at NA12878 because we always look at NA12878. She's kind of a rock star. So when we want to call variants, if we call variants against NA12878, we know in theory, because she's been sequenced so many times, we know what the value should be. As we build filtering models, we can run it and say, how well are we doing with joint calling? And for just a couple of samples, we find that we're pretty good about finding the known variants for her, but we miss some. Our sensitivity is not it is good, but we miss some. And you can see as you start adding more and more samples in, you can see that the curve goes up, that we actually gain more sensitivity. We start, as you, again, have more and more variants to actually look at. It's a little different between SNPs and indels. Indels are always harder, to be completely honest. They sometimes happen in messenger regions, but fine. But you can see, especially that with SNPs, that as you add more and more samples, it doesn't have to be a crazy amount. Not a crazy amount of samples. You start to get a lot of really good confidence. We calculated before, Laura, a really good colleague and very funny, around 600 samples, you actually start to have really good saturation. Like that's enough to train a model. For the most part, what is or is not a variant, certainly for common ones. And this is one of the ways, by the way, we'll talk about these later, like sensitivity and precision in terms of how we can measure how our joint calling is doing and just how our calling is doing in general. But it doesn't take that many to actually start getting much better data. Yeah. And again, just because you're joint calling, worth noting, you're joint calling and it'll give you a lot of information about like variants that are going to show up in the population. That doesn't mean you lose precision in finding variants that might show up in an individual. All right, you still find them. We can say that, again, it was measured. We find that we still maintain the ability to call singletons. That might be a de novo mutation on a particular person, or that might just be the only person in a sample that has that mutation. Okay, but we don't actually lose that by joint calling, which is to say that you gain a lot, you lose extremely little, beyond the fact that you have to do processing, of course. And it's important to note that also it can be noisy to have to go in and filter, to have to filter out a cohort and everything else. It can be, but the beauty with a lot of joint calling is that even if you have a variant in there that you're maybe less sure about, a lot of joint calling pipelines do soft filtering. They'll flag it and say, oh, I'm not sure about this one, give it a confidence score. It'll still be in there though, because depending on what you're looking at, you might want to look at those. So when you look, for instance, in here, like if you analyze these two samples individually, right? You can say that, well, you couldn't really get the call at either one of those important locations based purely upon the information in sample A, or for that matter, purely upon the information in sample B. Neither of them could give you a good call, but it's worth noting that in a pure VCF, you don't know why. You didn't call a variant, but you didn't call a variant for two very different reasons. In one of these cases, you had a lot of data and a lot of coverage, and you said, okay, well, I think it's ref because you were pretty confident that it was ref. In the second case down here, you said, well, I can't call a variant there, or I can't call a variant there, but not because you had a lot of confidence that it was referenced, because you didn't really have any good coverage there. Now, in a VCF, those two things might show up the same. You're not calling a variant, but you're going to want to know why. Because in one of these cases, if it's clinically significant, one, you might tell somebody, okay, yeah, in fact, these particular genes here are predictive of Alzheimer's. You want to be able to tell them either we're pretty confident that you do not have a mutation there, or you want to tell them, well, for whatever reason, our sequencer didn't cover that region. Maybe have it done again. They're very different clinical outcomes if you think about it. I don't know versus I'm confident you don't have a variant. You lose that just looking at a VCF, by the way. GVCFs maintain that information and we can use it in joint calling, but you lose it just looking at a VCF. So what does joint calling look like? Right? Well, logically speaking, what we tend to have here is that it's a two-step process. First, you look at each individual VCF, and like we did in our example, you use the haplotype caller on the individuals. It gives you calls. Specifically, you're also going to spit it out in GVCF mode. So you don't just have calls, the variant calls, you also have an analysis of the ref blocks and confidence in them. The second thing you're going to want to do is go in, consolidate all those individual GVCFs into one group somehow, and then what you're going to want to do is run joint or genotype GVCFs on them all together and use a multi-sample model. So that's the general two-step process we have when joint calling. First, you run them through the haplotype caller individually. Second, you spit out GVCFs, and then you consolidate those GVCFs together and you joint call them. Now, as we mentioned before, the thing about GVCFs is they got that funny non-ref block there. I mentioned last time, I mentioned again. That's how GTK communicates the fact that, oh, yeah, I think it's ref, but here's how likely it is to be something else. That's information you really need when you're going to analyze an entire population. Remember that case we mentioned a few slides ago here of potential Alzheimer's? If you're looking across the population, I mean, when you're looking at maybe statistics of mutation there, is one thing to say, oh yeah, that person's ref. They're fine. That's another count for ref. It's another to say, we don't know what that person is. We have no idea. So you can't count when you're trying to figure out how likely a variant is, you can't use it. You can't count it toward ref just so we can't call it variant. The truth is we don't know what it is. It could be anything. And that's information that the GVCF maintains. It still tells us, not just is there a variant, but do we really know what's going on? That is the benefit, by the way, of the GVCFs. Now, they're really valid VCFs. So I mean, VCFs have their normal blocks in the header. I looked at them a little earlier. You might have seen it there. I don't really recommend eyeballing VCFs a lot. I mean, it helps to look in there. It's just, I don't know. I always leave in a bad mood. I don't know. You got to look in it. And there are tools that also analyze it for you. But you see, a VCF has certain fields at the beginning. It'll tell you what the alt, the filter, the format, all information that the haplotype caller knew about that particular site. Well, the GVCF still gives you that. It gives you all that information. It just also throws in some new headers that will explain, for instance, the GVCF blocking. Remember, I mentioned that a lot of times for ref calls, if they're contiguous, it'll go ahead and spit them out of the block. It'll also do some grouping and say, well, if it's between 10 and 15, is that really so different? for a ref call, you care about that for variance, right? You want to know everything you can about variance. But for ref calls, oftentimes you don't necessarily. It's sufficient to be close to say how confident you are. You want to know if you're not confident, if you're pretty confident, if you're really confident, but we often do blocking, for instance, on those and that also helps us compress the ref data a bit and say, we have no confidence in that block. We're pretty confident in those. We're very confident in those, but the level of precision you need in reporting that isn't quite the same. So you have the GVCF block header in there as well, and that's also one of the additions. It's still a valid VCF again. It just has the extra stuff in there that have to do with how we actually spit out the data in our GVCF. So the thing is, even though GVCF files have that extra information, got extra headers, it's got that funny kind of non-ref token in there, when you actually joint call, like we actually do your joint call at the end, you still get a normal VCF, okay? We don't continue to have the extra information in there. The final version, like here you'll see, we have the ref block here at 204, another one down there at 212. Well, you have a variant call at 211. That's all that's going to come through. In your final joint call VCF, it's just the variant calls again. That information is used internally in trying to determine what variants to call. But at the end, you don't carry on that extra information with the non-ref blocks and all the reference sites to the end. Like that's only an intermediate format you use there. And again, we do have records, by the way. I mean, technically, there's also an argument. If you really, really want to get the non-variant records in your final one, maybe you need it for a particular use case. We don't stop you from doing it. There's documentation. We have documentation on our website that will tell you the arguments to do, because in some use cases, you might have to do so. So before you do joint calling, you have to find a way of bringing all your individual VCF together in GVCFs. In GATK3, well, it worked. There's a hierarchical merge there, and it goes through about 200 samples at a time. It outputs the GVCF with all the different columns in there. It functioned. It just didn't scale well at all. I am personally blaming Eric for that. Now, when we went through our exercise earlier, you note that we use genomics DB import, okay? That is the way to do it in GATK4. You pass in the arguments you want. It creates the genomics DB data store on the back end, and that's that. It just takes them all at once. It spits out its own internal representation. That scales a lot better. The thing to know about it is that it doesn't choose an arbitrary store on the back end. It specifically stores them in such a way where it can process them all efficiently in batches. Because when you actually do the joint calling, that can be computationally expensive. There are steps that can make it expensive when you're bringing them all together. I guess afterwards you can ask me if you really want to know the details about why. It's been entirely too much time. Now, the thing is, realistically though, as we mentioned, like in GATK3, you can only get about 1,000 genomes in there. It's about the most you could do doing that. GATK4, well, you can actually get up to about 200 or about 20,000. And that's actually whole genomes, by the way. Exomes obviously have a lot less data. You can get more of them in there. But with whole genomes, you can get up to almost 20,000. I'll point out something too, though. Let's say 20,000 is a practical limit. You can go above it using genomics DB. You can go above it if you like pain. And if you like frustration and pain, I am not going to stop you. You can ask Eric all about his experience trying to do 60,000 whole genomes in genomics DB. Ask him, maybe get him a drink first and relax him, because it is not, it's a very frustrating process. And we saw an example of this earlier, by the way, when we are actually running genomics DB import. We saw what it looks like. You pass in, you give it a path, a genomics DB workspace path, you pass it a series of arguments, it goes ahead and creates a data store for you. Now, it's worth knowing that by the time you get down to genotype GVCFs, it can either take a single GVCF that has been merged together to produce a lot. And we saw that, by the way. Remember when we were looking at what was in genomics DB, we actually had it, we called select variants and had it spit out a GVCF so we could see that it had data for all the samples in there. You could pass one like that into genotype GVCFs, but really you probably want to pass in the path to the data store. You know, it can go in and it can actually just directly read from a genomics DB data store, and that's a lot better, to be honest. It's a lot faster. So here, you can see what it actually looks like. If you're having a single or multi-sample GVCF, yeah, you pass it in -V, and you give it just the path to the VCF. or the GBCF. But as you saw before in the example we just went through earlier, you can also pass to that GenDB path. Okay? That GenDB path gives it a location of where you built your genomics DB data store, and it can go in and pull them out of there. It can work with them in that format, which, like I said, it's a lot more efficient to work with it there. So, like I said, in the GTK3 way, well, you can do consolidate GBCFs. You combine them 200 at a time into one giant multi-sample GBCF, and you kind of spit them out and pass them into, I guess, genotype GBCFs, and it works. But the truth is, GTK4 with Genomics DB is just a lot better. Okay? Now, using that, we're able to produce the whole Nomad V2 and the CCDG joint call sets. It went up to 60,000. I'll put a big asterisk beside that because it took a lot of manual knowledge and intervention to do that. I believe Eric actually produced some of those big call sets there. He gets a 1,000-yard stare and a haunted look when you ask him about them, but he was technically capable of doing so. They took about 3 weeks to do, so you can, with enough knowledge. Either way, you've seen about a 20-fold increase in scale. That's needed. The truth is, at the time you mentioned writing GTK, there weren't that many sequence files in the world. That has changed, and it's still changing. And there's a lot more data out there, not just because, just for disease purposes, but there are all these large biobanks. And the size you have to be able to operate on is changing now. So, don't use it. Okay, pretty much you have almost no reason to do the GTK way, the GTK3 way. Like, the truth is, Genomics DB is better essentially every possible way over what we were doing in GTK3. All right? But here's the thing, though. And like I mentioned, the numbers keep getting bigger. 60,000 with some pain might sound like a lot, but the UK Biobank, that was about 470,000 exomes, not genomes, exomes, but that's still a lot. The All of Us initiative, that's a US National Institute of Health program just to sequence up to 1 million people in the United States. 1 million whole genomes, too. Most of you here work with genomic data to the degree where you understand how large a lift that would be. It's a huge ask. Genomics DB, it's not going to cut it there. It'll get you up, again, practically speaking, to 20K, or maybe with some pain to 60K, but that's not where you're going to go. So the truth is we have been planning for the future a little bit more. We have a new next-gen product called the Genomic Variant Store that actually lets you enable joint calling for extremely large cohorts. It works just fine on small cohorts too, by the way. I regularly run it on 3 or 4 samples. But that is our next generation project. It's really designed to handle the future of these massive biobanks. Eventually, we're planning on scaling it the whole way to 1 million. That's what ALU asks, and we want to be able to handle call sets like that. So for instance, the All of Us initiative, all of their giant call sets, we use this tool, the Genomic Variant Store. We use GVS for those. We did 98,000 whole genomes back in March and June of 2002. We did 245,000 whole genomes. That was released in April of this year. And I can say right now, we're actively working on their 380,000 whole genome call set now. This is our ramp up, basically, Genomic Variant Store. Right now, though, it's in a limited access beta. Why? Because it takes a lot of work to continue scaling, believe it or not. Even jumping from 250,000 to 400,000 takes iterations, changing our data store, changing algorithms, stuff like that. So we do have a closed beta or a limited beta that people can get access to. Don't worry about the link there. You can click it, but the link is available in the actual PDF version here. It's built on the Google Cloud, just like Terra is. Right now, it uses BigQuery for its backend. I mentioned that Genomics DB actually goes in and creates a special format. It has a Genomic DB database. Well, you're doing a very, very similar thing here, but you're actually using Google BigQuery product as your backend. It's very fast. We are able to get some impressive numbers I'll share with you soon. And it also, because it's using BigQuery, it allows you to do some arbitrary querying over your data. Genomics DB doesn't really allow you to do that. It pulls all the data in, but it's somewhat opaque. In GBS, you can just query over the data and answer some pretty important questions. I'll go into that in a little more detail in a slide or two here. And when you do that and you run your joint call set, it will still spit you out whatever you want. It can spit out a big joint called VCF at the end in pieces because it would get too large. It'll spit you out a sharded VCF, or for that matter, it can also spit out HAIL VDS files. HAIL is one of our tools for analyzing large amounts of data as well. Again, the VCF format only gets you so large. For doing 250,000, we didn't produce a 250,000 sample VCF. That would be bad. So there's another format too, where you can go into HAIL. And at the VDS format, you can open it there. Also, we're doing Link PGen format as well. So despite using its own data store in the back end, it can spit out data in pretty standardized tools. So what it's doing is not that conceptually different from Genomics DB. Mostly it has to do with that consolidate GBCF step. Genomics DB, okay, it processes them, gets up to about 20,000. You can run Genomics DB import. GBS actually works in Terra workflows. We spoke about this a little bit earlier, that using the workflow description language and running the pipelines there. Yeah, to get up to that level of skill, running them individually, you can do some, but realistically controlling exactly what the machine size is, how much memory to allocate, different steps have different load, different types of load, different characteristics there. We run this inside of workflows. All right, so that's a Terra pipeline, although it can run with some help in other pipelines other pipeline runners as well. And an important thing is that GBS also lets you add in individual samples later. One thing you see with these massive call sets is not uncommon for them to say, okay, that's great. Oops, can you remove these 27 samples? You don't want to have to start the whole way over. You don't want to start over and say, I guess I'll re-import another 250,000 at that point. No, people would cry and probably get fired. So it's worth noting that GBS does a little more than just Genomics DB does as well. It actually replaces more of the pipeline. Again, I mentioned Genomics DB's data store is not searchable. The one for GBS is. It covers the ingestion, like what you might see there when you did Genomics DB import. It covers that part. It also does variant filtering. We'll have some slides, I believe tomorrow is there, where we cover the filtering models. It handles VQSR, our classic one, as well as VETS, the next one. Feel free to ask after about them if you want to know details of those. And it does a joint calling. And also, importantly, it does allow data store to be searched. Because it's fundamentally using BigQuery for its data store, you can search it using SQL. If you know how to write SQL, you can just write SQL queries. Like, find me all participants with a variant at that position. Oh, you can do that. For instance, some of the people doing analysis on the AOU call said, he's like, yeah, I ran it, came back in a second. I got my answer. Like, BigQuery can be very fast for these kind of large-scale computations. Also worth noting, I think Eric mentioned earlier that there's other information you can use when you have these large call sets. You can annotate variants about parts that are clinically significant. Using various databases that collect that. There are auxiliary pipelines along with GBS that let you do that. That will take the variants in a call set and tell you, like, here are the ones that we pulled from various annotation products, and here are the ones that were clinically significant from it. And that's information that can also be searchable for. So really, GBS does everything Genomics database did, but faster and better, to go on for the next generation of big call sets. And to get the numbers down, so running through GBS, when we have 1,000 genomes, it took about 8 hours, less than $100. What we're seeing is like about 6 cents a sample. Now you can see as we keep scaling up, that price only goes up a little bit. By the time we got to almost 100,000, that was a 2-week call set, went up to about 8 cents a sample. That was mostly because, by the way, VCFs don't scale linearly. We expected we'd hit an inflection point with VCFs where they'd start going up and up and up. Again, I blame Eric. That's okay. Between that one and the 245,000 call set, note the time. Still took about 2 weeks. And we actually got the cost per sample down. And we expect that to stay flat or even keep lowering. I mentioned we're spitting out something called a Hell VDS file. It's useful for opening in Hell, but not writing a VCF allowed us to get under that cost curve again too. So it's pretty cheap, it's pretty fast, even for pretty small call sets. Now, like I said, it's in a closed beta, but we've seen a lot of success with beta users. Like one of our beta users doing some research on ALS, actually, was kind of funny. He came back and basically told us, he's like, yeah, I told my colleague when it was done, and his mouth dropped. He's like, really? Really? You could finish that in how many hours and it costs what? So we do find that a lot of times the numbers, even for like small call sets, are, we have to convince people they're really true. Now, I point out again, GBS might be in a closed beta right now, but we're actually targeting release of it to be next year. We're trying to target maybe fall of next year. There are a couple of things we want to add in, specifically some stuff around exomes, possibly different references, stuff like that. But we're aiming for about next year. For now, it's a limited beta. You can sign up for it. But yeah. Now, once you've done that, once you've actually done your big junk call set, the next steps are, well, filtering your variants is one thing to have them all together, is one thing to say you have the data to tell what might be junk and what might not be, but you have to actually do it. And now if you're running GVS eventually, it actually does that as part of its pipeline. But since it's closed beta, we'll go over how you do it with the existing GTK tools, okay? And after that, we didn't have a chance to demonstrate it much earlier, but there's also variant refinement. Again, the same thing, you can sometimes pull in outside information to get a better call on there. In the particular example we didn't get to, which you can go through on the notebook, you'll see we can pull in relationship information so that if you can tell the tools, hey, these two are the parent of this one, it can go in and figure out what calls might be in regions that are there. You can pull in population statistics, and it can use that information to further refine variants. There are a bunch of cool things there. I do recommend you finish going through the bottom part of the notebook we ran out of time for, but that's really where you want to go next after you've done joint calling. So actually done with the joint calling slide. I believe I've run right up to the end of our time today. However, have any questions? Well, Eric might also be coming to steal the microphone from me. So
I'll say that the instructors will remain around for the next 10 minutes or so. So when everyone is dismissed, feel free to stick around and ask us questions for a bit. And we'll be like tomorrow morning and tomorrow afternoon. There's plenty of time to ask us questions.